Okay, so thank you very much. I'm here talking about development of anticoccidial vaccines. This is work we've been doing as part of the SAPHIR project. So it's fair to say chicken production is booming. More than 64 billion broilers were produced in 2016. So there's vast numbers of chickens out there. Well, the map you can see here is a heat map of where the demand for chicken meat is going to be in the coming years. So you can see as it moves to the darker red areas are the regions with the greatest increasing demand for broilers. So you can see there's increasing demand in Southeast Asia, North America, South America, across North Africa, Sub-Saharan Africa. But of course, as we increase the number of poultry, we're also increasing the risk of pathogens that infect poultry. And we're particularly interested in Eimeria parasites. So there's 7 Eimeria that infect chickens. And as far as we can see, wherever you find chickens, you are likely to find one or all of these species. So we can say that if chicken production is booming, then so is coccidiosis. And that's the disease that's caused by Eimeria. Now, if we look at the figures that underpin this, well, the global cost of coccidiosis has been estimated to exceed $3 billion a year. And this is a combination of the costs of the disease, so losses caused, but also the cost of controlling the parasite. Within the UK, we see that about 40% of all drugs used in agriculture to control pathogens are used to control coccidia. So this is actually a massive proportion. So if you could find another way of controlling Eimeria, then you could have a dramatic reduction in drug use. Now, a combination of these two figures has seen coccidiosis ranked in the top 10 economically important endemic veterinary diseases in the UK, largely because of the fact there's just so many chickens produced. So pathogens that affect chickens have a very high economic impact. And they've also been ranked in the top 10 veterinary diseases or pathogens in Southern Asia, and that's based upon impact on the poor. I think the point here is if you can't afford cattle or pigs or sheep and you're relying on poultry, then pathogens that affect poultry can have a devastating impact. So we know that Eimeria have been around for a long time, but they're not going away, and control remains absolutely essential. Now, control has historically been based on the use of drugs. There's a range of anticoccidial drugs. And you can see here in this graph, we're looking at the chicken population in the world. And actually, from the 1940s, it's been on a very steep increase. We've been increasing poultry production, increasing intensification. And this has been supported by the appearance of drugs. So we started off with the sulfur drugs that we can use to control Eimeria. They're followed up by a panel of synthetic drugs. And after that, there are ionophores. But the key point is there's been no new drugs produced that are released to control Eimeria in the field since the early 1990s. Now, we still rely on these fairly old drugs. We know that resistance is very common to all of them. It develops very quickly, it spreads very quickly. And the only way we can control Eimeria at the minute is by strategically either combining drugs or rotating them. And this does work reasonably well. However, there's increasing pressure on the use of drugs, both from the general public, from the commercial sector, and from legislators. There's problems with food contaminants, there's problems with resistance, there's a general drive to reduce drug use in livestock production. And there have been a range of points where this has been pressure tested. So in 2008, the European Union launched a report on the use of anticoccidial substances as feed additives. And the idea behind this was to decide whether to ban some or all anticoccidials in the European Union. In the end, that didn't happen. There wasn't a ban, I think largely supported by our experience with Histomonas, where they did ban the drug, and a pathogen that was well contained has become much more of a problem again. But alongside this, we see in the US, the FDA is starting to introduce restrictions on certain drugs in livestock production. And more recently, there's been the emergence of the no antibiotics ever branding. So this is where a range of companies, Purdue Farms, for example, McDonald's, Kentucky Fried Chicken, are demanding livestock products, so demanding meat that have been produced without antibiotics. And this is a real watershed change from a country that used to perhaps lead the way in the amount of drugs that was put into livestock production. And again, around the same time now, 2016, the EU has launched a new consultation to consider whether they'll revise the guidelines for anticoccidials. So if we're going to remove or reduce the amount of drugs we use, how can we control Eimeria? Well, there are vaccines available. They're very good vaccines, but they're not perfect. So in the US and in much of the world, we use wild-type anticoccidial vaccines. These are live parasites. You deliberately expose birds to these live parasites, normally at very low doses, and you allow the parasite to recycle through a flock, and it will stimulate a natural protective immune response. But the problem is we can only produce these vaccines in chickens. We can't grow them in cell culture, you can't grow them on plates. And that means there's an inherent limit to the production of these vaccines. So traditionally they've been restricted to use with breeder stock, with layer stock, because they're more valuable. But there is, as I say, quite a change. In the US, this reduction in drug use has seen a dramatic increase in the use of live vaccines. Here in Europe, we use attenuated vaccines. We don't, strictly speaking, have any registered wild-type vaccines, although they have been used on occasion. These attenuated vaccines have been selected for precocious development, so they have a faster life cycle, they produce fewer progeny, and they're less pathogenic, which makes them much safer. But it also makes them even harder to produce. And so there's a severe limitation on the capacity for this. Now, don't get carried away. There's still more than 1 billion doses of live attenuated vaccines sold every year. But if you remember, we said there's more than 64 billion broilers out there, then we need to find ways in which we can scale up vaccine production. So what are we going to do in the future? Well, the success of both live wild-type and attenuated vaccines is very clear. So there are going to be more of these vaccines produced. But there's also demand for cheap, effective, and scalable alternatives, especially so we can move to vaccinate the majority of the broiler sector. Of course, options to do this may be recombinant proteins, DNA vaccines, vectored vaccines, either dead or alive. But all of these rely on us knowing the right antigens to include in them. Now, fortunately for Eimeria, there's been quite a lot of work in this in recent years. We have some examples here. There's apical membrane antigen 1 or immune map protein 1, both of which are robust vaccine candidates. And if you look to the literature, there's actually loads of vaccine candidates out there. There's a range of micronean proteins, surface antigens, gametocyte proteins, all of which have some evidence that they can induce protective immunity. But we need to know how we can prioritize these. So if we just take one specific factor, I'm looking here at the ability of a recombinant protein to induce a reduction in parasite replication. And that's measured in terms of oocyst output compared to mock vaccinated. And you can see that each of these proteins, so we've got AMA1 and IMP1 I've mentioned here, looking at perhaps high 60s, mid 80s% in reduction of parasite replication. The others perhaps not quite so good. We've got micronean protein 2 and 3 around the 50% reduction, profilin, and a few surface antigens. So they're all effective, but I think it's very clear that these are not going to be the answer on their own. We're not going to have a vaccine based on a single recombinant protein. Now, we can put them in context. If we look at ionophores, and ionophores are the market leader in terms of control of coccidia, and one of their strengths is that they do give incomplete control. So one example, monensin, when it was first released, in completely drug-susceptible parasite populations, for Eimeria tenella, still only managed to reduce parasite replication by between about 75 and 95%. So even then, even with a new drug, there was some parasite escape. And you can see one of our recombinant proteins just manages to sneak into this same range. But reliably, I think it's clear that none of these antigens alone will work. So part of the SAPHIR project was taking these recognized antigens and combining them so we can then produce more robust, more reliable vaccines. But the biggest challenge with poultry is how are we going to deliver a vaccine? No farmer is going to go and inject every broiler in his flock 3 times with recombinant protein. It's just never going to happen. So we need to find an effective way of delivering antigens for vaccinating broilers. So what considerations do we have? What's important? Well, obviously, from a commercial perspective, they have to be effective. They have to stimulate an effective immune response. And as I say, for broilers, we require a simple delivery mechanism. So vectored vaccines are particularly appealing. Now, if we're looking at a vectored vaccine, we think there's some additional considerations. That vectored vaccine should be host-specific. We don't want to release it to vaccinate poultry and find it replicating in the rodent population or infecting humans. Ideally, it should be highly immunogenic. So if our vaccine vector can offer an adjuvanting property in addition to the antigens we provided, it'll be very helpful. It should certainly be non-pathogenic. We don't want to see any problems caused by the vector on the host. Ideally, it should be self-limiting, so we can control release of this parasite, of this vaccine. And it should also have the capacity for multivalence. Now, we saw a minute ago that we will need more than one antigen to be in this mix. Therefore, ideally, we should have a multivalent vector. Now, there's already viral vectors used in poultry production, but this notion of multivalence is going to be challenging. You cannot produce viral vaccines at the moment that include multiple antigens. So it would have to be a cocktail of different viruses. The self-limiting aspect's challenging for bacterial vectors, Salmonella's suggested, for example. So the work we're doing is thinking, well, can we take live Eimeria vaccines, but actually genetically modify them so that these transgenic Eimeria can express additional antigens and induce additional immune responses? So how do we make a transgenic parasite? Well, we start off with a construct. So this is essentially a DNA plasmid. It contains the gene that we're interested in, so it's our vaccine candidate, and it's combined with some controlling elements, stop codons, start codons, things like that. And we need a cell. That's the parasite. We don't use the parasite egg, which is called an oocyst, because it's too tough. So instead, we purify these things here called sporozoites. That's the first motile stage of the parasite's life cycle. But for this, we can consider it to be a general cell. So it has a membrane, it has a nucleus. And we can apply an electric shock, we can drive this plasmid into the nucleus, and this will lead ultimately to a transgenic parasite in this case. The ability to do this transiently was developed quite a long time ago, and to actually produce stable transgenic Eimeria was reported 10 years ago. So the notion is now, can we take this to develop Eimeria's vaccine vectors? I've been trying to do this over the last couple of years, and there's been some promising stories. For example, we can take an antigen of Campylobacter jejuni, this is CJA. You can create an Eimeria parasite that expresses the Campylobacter antigen, infect chickens, and you can generate a protective immune response against colonization by Campylobacter. In a second example, we've taken Eimeria parasites and expressed some viral antigens, either infectious bursal disease virus or infectious laryngotracheitis virus. And control of these viruses is all about antibody titer. If you have a high circulating antibody titer, you can control the virus, and we've shown that you can actually produce specific antibodies using this system. But our challenge here is to control Eimeria. So the idea is if we can take Eimeria oocysts like this, we can actually engineer them so they express additional anticoccidial vaccine candidates. And so we can then vaccinate chickens so that they don't look like this. They're protected from the parasite. So within SAPHIR, we've used a series of different strategies to try and produce these transgenic parasites. Now, to simplify them, we're looking to express our antigen, and the example we've taken here is apical membrane antigen 1. And we either express them within the parasite, and we can see here these are actual pictures of the parasites, where the green is a transfection control that shows you have a transgenic parasite, and the red is actually fused to our vaccine candidate. So everywhere you see orange, you've got an overlay of red and green, and both proteins are being expressed. We have a secreted form, and that's where the parasite produces the protein but then secretes it outside of the parasite. We chose this because it might be more effective at stimulating a protective immune response. And you can see there's a different profile now of where the protein is being produced and is ready to be excreted. And the last option we have is a surface tethered option. So this is essentially the same as our secreted parasite, but now instead of just releasing the protein outside of the parasite, it binds it to its surface. The idea being that as the parasite moves through the host, it will be waving that protein around on its surface, and it's able to stimulate a protective immune response. So I'm just going to report here the very first vaccination studies we've done. Now, in our first study, We wanted to see whether we could generate protection using these parasites. So what we did is we took batches of chickens and we gave them very low numbers of our vaccine oocysts in a trickle-type infection. So we're trying to mimic what would actually happen to a chicken in the field as a parasite vaccine would recycle. And we then challenged these birds with a low virulent parasite challenge to see if we could protect. So the readout we have here is parasite replication, and that's terms of oocysts being produced. We have down here our 3 vaccine options, so there's the internal, secreted, or the tethered vaccine types. We've put the vector in alone, so this is empty without the AMA recombinant protein, with PBS controls. And you can see when we vaccinate with each of these groups, we then challenge with the parasite that donated the AMA, that's Eimeria maxima, that in the PBS group, this is our control, there's a high level of parasite replication. There is some reduction with the empty vector. And this is quite promising. You know, if you think back to my list of vaccine considerations, we wanted an adjuvanting effect. So there is actually a vector effect here, a reduction in parasite replication. But then when we put our transgene in, it reduces even further, between about 45 and 55%. Interestingly, there wasn't a significant difference between our 3 different vaccine systems. So it looks as if we could take, at this point, any of them forward. Now, of course, that's just one study. We like to be a bit more confident that something actually works. So we repeated the study, this time just using the internalized version. But here we either vaccinated birds once or we vaccinated them 5 times. The 5 times being, again, that notion of parasite recycling in a flock. We have the empty vector as a control, and we have PBS. And again, you can see in the PBS vaccinated birds, so not vaccinated at all, there's high levels of parasite replication. Where we've had the empty vector, there's modest levels or modest reductions in parasite replication. Again, this notion of cross-protection or an adjuvanting effect. But with either a single or multiple rounds of vaccination with the test vaccine, there's significant reductions. And you can see in all of these cases, we've had from 47, 38% here, that's a single vaccination, up here to 45, 55, 50% reductions. And for this antigen, that's what we would hope to see, where we use it as a recombinant protein, use it as a DNA vaccine. So it's very promising. Now, we wanted to go forward because, of course, whilst parasite replication is important, what a farmer's interested in are production traits. So we're talking about weight gain in broilers, we're talking about whether a bird actually gets sick. And as a start, just emphasize we were looking here at either a single or multiple doses. So to test it, we started off by just looking at a single vaccination event. So I want you to bear in mind these figures are erring on the side of caution, that we've just had a very low level of vaccination. So the first point, we're looking at protection in terms of body weight gain. So if you have birds that are PBS, so they're not vaccinated and they're left unchallenged, they have a high level of body weight gain. If we compare them to challenged birds, so these are birds that are unvaccinated but exposed to the parasite, then there is a significant reduction in body weight gain. We're given a moderate parasite dose, so we're not expecting to get sick, but there is a production effect. Now, when we look at what you would imagine as a traditional vaccine would be, so this is where you've given them a live parasite to begin with and then challenged them, you'd expect to have good protection here, and again, it has because the figures are quite high, the birds are performing well. When you look at the tethered vaccine, which we have here, so this is where we've actually got our AMA1 antigen, it's been expressed, and we can see that the parasite body weight gain, the chicken body weight gain is quite high, and it's higher than you see in the empty or the mock vaccine group. So the take-home message here you have is that there was a moderate parasite challenge, so we've got a reduction in body weight gain, a normal traditional live parasite vaccine protected against it, our transgenic vaccine protected against it, but the vector alone did not. And to emphasize the point, this was with a single vaccine challenge. If the vaccine was to recycle, as we saw before, we would expect to have better levels of protection. The same birds we looked at for lesion scoring, and you can see that in birds that were unchallenged, well, they've got no lesions, as you would expect, there's no parasite present. If they've not been vaccinated, they have reasonably high levels of lesions, between 2 and 3. Bearing in mind a lesion score of 4 is a bird that's severely sick, perhaps maybe close to death. When we're given a live parasite, so that's akin to the current live parasite vaccines, there's good protection. There's no protection at all from the empty vector, but when we add the AMA antigen, we do see a reduction in lesion score. Now, it's not equivalent to a live vaccine at this point, so it does emphasize the fact that we will probably need more antigens, and again, we will be relying on more vaccination events, but it's a very promising start. So to summarize, development of anti-parasite vaccines is immensely challenging. Parasites have evolved to escape immune responses. That's their whole purpose. There's many parasite targets, but there's very few commercial vaccines, and at present there's no subunit vaccines that are appropriate for use with broiler chickens. There are a range of vaccine formulations available, and of course our challenge is that a future subunit vaccine must be effective, it must be safe, it must be cheap, and it must be easy. And for broilers, those last two are absolutely essential. We've shown that Eimeria can function as a vaccine vector, but it requires some more development, specifically looking at the right antigens to put into the mix, and we accept that a combination of antigens will be required. Now, the studies we're working on at the minute are actually testing different combinations of vaccines, and that's something that we'll be talking about perhaps in the coming year. So the idea is that one day we could go from products such as Paracox 8 down one day maybe to Paracox 1 with a single vaccine that would express antigens from multiple parasites. So just to finish up, I'd like to acknowledge Ivan Pastor-Fernandez. Ivan's done the majority of work that I've spoken about today, supported ably by several people at the Royal Veterinary College, and of course the funding through SAPHIR. Thank you very much for your attention.